Assalamu alaikum. Welcome to our channel Botany with YouTube. Hope you all are fine. I'm here, a PhD scholar. I'm going to present a topic, transformation. Genetic material in the form of naked DNA is transferred between microbial cell. You can see here in diagram transformation process in which bacterial cell and bacterial chromosome is transferred from one cell to another cell. Bacterial transformation. Transformation, type of genetic recombination in bacteria. Transfer of free DNA released from a donor bacterium into the extracellular environment that result in assimilation and usually an expression of newly acquired trait in a recipient bacteria. Sexual reproduction is absent in bacteria. If you are new in our channel, then please try to subscribe our channel. Firstly, discovery. You can see here scientist picture. He was a British microbiologist who discovered the transformation process through his experiment on pathogenic bacteria Streptococcus pneumonia. Frederick Griffith's experiment used two strains of Streptococcus pneumonia bacteria, for example, smooth strain. It's also called normal form, smooth clunies, virulent, and the other type of bacteria which he used in his experiment was synthesized a polysaccharide capsule caused death of the host, for example, rough strain, also called mutant form, rough clunies, non-virulent, no polysaccharide code is present. Frederick Griffith experiment. You can see here in first diagram R strain, non-virulent, inject mouse with R strain, mouse injected with R strain lives, R strain virulent. In this, inject mouse with S strain and mouse will be dead. Heat killed R strain, inject mouse with heat killed S strain, mouse injected with heat killed S strain live. Then at the end, fourth point, heat killed plus R strain, inject mouse with mixture of heat killed S strain plus R strain. Mouse injected with mixture of heat killed S strain and R strain will be die because heat killed and R strain both have ability to transfer their DNA from one body to another. Griffith concluded that sometime in the heat killed S bacteria transferred the hereditary properties of R bacteria. The nature of this transforming principle was unknown. Transforming principle. Oswald, Avery and his colleagues performed experiment which is called transforming principle. You can see here scientist picture. He and his colleague identify which factor involved in transformation. They start biochemically removing the things until the transforming power is lost. In his experiment, he prepared mixture of dead S and live R streptococcus. Remove the protein, then RNA, then DNA. Only the final DNA treatment destroys the capacity of S to transfer R. This indicates transforming substance as DNA. Steps of transformation. Number 1. Competent cell preparation, transformation of cells, cell recovery, and cell plating. You can see here in diagram bacterial transformation, competent cell preparation, transformation of bacteria, recovery of bacterial DNA or genetic material, and plating. Mechanism of transformation, and this first step is competence, the ability of a cell to alter its genetic by taking up extracellular naked DNA from its environment. Types of transformation. Number 1, natural transformation. Number 2, artificial transformation. You can see here the logo of our channel. Then please try to share our channel with your friends, with your students, as well as your colleagues and other researchers for getting valuable information related to botany course outline or related to botany subject. Approximately all the syllabus of botany paper A, B, C, D and MS PhD are also available here. Natural transformation discovered Frederick Griffith in 1928. Incompetence factor vary among genera. Protein produce homology different in gram positive and gram negative bacteria. Once the DNA has been brought into the cell cytoplasm, it may be degraded by nucleases enzymes, or it may be DNA repairing enzyme may recombine it with the chromosome if it is very similar to the cell own DNA. You can see here the diagram. Recipient cell take up donor DNA and then recombination occur between donor DNA and recipient DNA. Genetically transferred cell form. Artificial transformation. In this procedure, cells are made permeable to DNA. Two methods of preparing cell competent: calcium chloride and electroporation. Laboratory procedure, simple and easy method used in genetically engineered in bacteria, but in general transformation efficiency is low. Artificial transformation, you can see here in diagram, antibiotic sensitive bacterial cell, calcium chloride treatment is provided, add plasmid DNA, transferred bacterial cell, selection on bacterial growth medium containing appropriate antibiotics. Please try to like our video and share with your other friends and colleagues. You can see here calcium chloride method and electroporation method. First one is calcium chloride method. In this, cells chilled in presence of calcium chloride make the cell wall permeable for plasmid DNA. Cell incubated with the DNA, then heat shocked for 30 to 120 seconds on 40 degrees centigrade. DNA entered in the cell. And second method is electroporation method. Make hole in cell. Cell are shocked with electric field 100 to 200 voltage. Through holes, plasmid DNA entered in cell. After this, natural membrane repair mechanism close these holes. You can see here the chemical transformation and electroporation transformation. Chemically competent cells, incubation, heat shock, recovery, and chemical transformation process. And electroporation method includes incubation, electric shock, and recovery. Application of bacterial transformation. To make multiple copies of DNA and DNA cloning, to make large amount of specific human proteins, for example human insulin, which can be used to treat with type 1 diabetes, to genetically modify a bacterium or anther cell. Conclusion: DNA carrying hereditary information and it is involved in transformation process. The goal of transformation is to produce multiple copies of a recombinant DNA molecule. Thank you so much to all of you. Thanks to visit our channel. Thanks for your concentration. Thanks for your attention. Good luck to all of you. Allah Hafiz.